Hello and welcome, dear participants. Thank you for joining this pre-recorded webinar. On behalf of Pharmaskillcraft, we truly appreciate your time and your commitment to continuous learning. Before we begin, please make sure you are seated in a quiet and comfortable place. Also, kindly check that your internet connection is stable for a smooth experience. Since this is a pre-recorded session, we request you to post your questions or comments in the comment box. Our team will be happy to get back to you with answers. Your webinar hosts are Dr. VP Sutar and Mr. Sunil Patil. We hope you enjoy the session and find it very useful.
Hello everyone, and a very warm welcome from Pharmaskillcraft. Today, we're exploring something every pharmaceutical professional should know, the Burkholderia cepacia complex, or BCC. It may sound harmless, but it's a silent intruder in the world of non-sterile manufacturing, tough to detect, hard to kill, and capable of serious patient harm. So, by the end of this session, you'll understand exactly why this bacteria matters and how we can detect and prevent it. Let's dive in and uncover the hidden world of BCC contamination. Here's the big picture. BCC isn't just one microbe, it's a group of closely related bacteria naturally found in soil and water. They form biofilms, sticky, slimy layers that protect them from cleaning and disinfectants. Now, for patients, especially those with cystic fibrosis, low immunity, or in ICUs, infection can be severe. And look at this, BCC loves water-based pharmaceuticals like syrups, mouthwashes, and saline. It even survives preservatives that are supposed to stop bacterial growth. So tough bacteria, tough consequences. That's why detection is so critical. Let's travel back in time for a moment. 1949. Walter Burkholder first finds this bacterium in rotting onions. That's where it all began. 1950s. Scientists realize it can infect humans. 1977. First isolated from cystic fibrosis patients. That's when its medical relevance became serious. 1980s onwards. Pharmaceutical contamination cases start increasing worldwide. So, from a farm pathogen to a hospital and manufacturing challenge, BCC's story spans decades. Interesting, isn't it? From onions to ICUs, quite a journey. Now, why is this bacterium such a menace? Three main reasons. Antibiotic and preservative resistance. It survives most standard antimicrobial agents. Affinity for water. It thrives in aqueous formulations like syrups and saline. Delayed detection. It may pass QC tests, then appear later due to slow, uneven growth. In short, it can sneak through even well-designed quality systems. So, imagine a batch that looks fine today but fails next week. That's the nightmare BCC can cause. Let's look at real incidents to see the impact. 2016, liquid docusate recall, multiple pediatric infections, especially among infants. 2019, hospital nasal spray outbreak, spread within ICUs. 2022, FDA recall of antiseptic solutions contaminated with BCC. These events led to severe infections, extended hospital stays, and, tragically, patient deaths. So, BCC contamination isn't theoretical, it's a real, documented threat. How does it sneak into the production line? Through poor hygiene or unclean equipment. From biofilms in water systems, which act like bacterial homes. Or via contaminated raw materials entering at the start. Once it's in, it's incredibly hard to remove. That's why we say, prevention at the source is far more effective than trying to fix it later. Here's what regulators say. The FDA and USP require strict microbial control in non-sterile products. USP Chapter Number 60, introduced in 2019, specifically targets Burkholderia cepacia complex. It provides an official method for detecting BCC in non-sterile formulations using selective culture media. So, compliance with USP Chapter 60 isn't optional. It's essential for safety and regulatory approval. This chapter has changed how the industry handles microbial testing. To meet these standards, facilities must follow rigorous hygiene practices across all operations. Design and monitor water systems carefully to avoid biofilm buildup. Validate test methods specifically for BCC. Perform in-process quality checks, not just end product testing. And investigate failures immediately with CAPA actions. Together, these steps form a safety net against BCC contamination. USP Chapter Number 60 was officially published on December 1, 2019. Its title, Microbiological Examination of Non-Sterile Products, Tests for Burkholderia cepacia complex. What is the goal? To verify that products are free from BCC. This standardized method gives manufacturers a clear, reproducible way to detect contamination before release. Now, let's see how the test works. We collect a product sample, dilute it, and incubate it at 30 to 35 degrees Celsius for up to 48 hours. Then we plate it on Burkholderia cepacia selective agar, or BCSA. BCC produces distinct greenish-brown colonies with halos, thanks to pigment called as pyocalin production and pH indicators. Meanwhile, antibiotics like polymyxin B, gentamicin, and vancomycin in the agar block other bacteria, so only BCC grows. That's the brilliance of selective media. BCC stands out clearly. Here's the medium recipe. Nutrients: casein peptone, yeast extract, lactose, sucrose, NaCl. Selective agents: polymyxin B, gentamicin, vancomycin, crystal violet. Indicator: phenol red for pH change. Together, they create conditions where only BCC flourishes and competitors are suppressed. So every plate becomes a targeted environment for BCC detection. Each medium batch must be validated using reference organisms. Here is the table of some BCC strains used for testing of media. To check selectivity, non-target strains like Pseudomonas aeruginosa and Staphylococcus aureus are also tested. If BCC grows and others don't, your medium passes. Let's quickly walk through our validated process for culture suspension preparation. It's all about control and timing. We start by taking our test strains and inoculating them into or onto soybean casein digest broth or agar. We then incubate them at 30 to 35 degrees Celsius for 18 to 24 hours. This time frame ensures we get actively growing, healthy organisms. Next, we harvest that growth and place it in a tube with a specialized diluent. We must use a gentle, buffered solution, either buffered sodium chloride peptone solution, pH 7.0, or phosphate buffer solution, pH 7.2. These buffers keep the organism stable and viable. Now for the critical part. The clock starts immediately. For the most reliable results, suspensions must be used within 2 hours. If you need to store it, keep it refrigerated at 2 to 8 degrees Celsius, but you must use it within 24 hours. This is the hard deadline for your validated method. Commercial kits. If you're using ready-made suspensions or bioballs, simply follow the supplier's instructions. They've done the validation for you. Let's understand how we can test BCSA for GPT properties. We begin by inoculating the new plate with a very small, challenging number of the test organism, not more than 100 colony-forming units from the suspension. This low count ensures the medium is genuinely tested for its ability to promote growth. The plates are incubated under controlled, standard conditions, 30 to 35 degrees Celsius for not more than 48 hours. Using these exact, verified parameters ensures that any variation in the result is due to the media, not the environment. The final step is the critical acceptance criteria. The resulting growth must be comparable to the growth previously obtained on a tested and approved batch of medium. If the new batch performs as well as the standard, it passes. If growth is weak or absent, the media fails and must be rejected. Test for inhibitory properties of BCSA. BCSA is a selective medium. Inhibitory testing is crucial to ensure the medium correctly suppresses the growth of unwanted microorganisms. To perform this, you must challenge the medium's selectivity by inoculating it with at least 100 CFU of the appropriate test microorganism. As specified in Table 1, the microbe the medium is designed to inhibit. Following this, the plate is incubated at the specified temperature for not less than the longest period of time required by the test protocol, ensuring the inhibitory agents have ample time to act. For the medium to be accepted, the acceptance criteria must be met. Inhibition of growth of the indicated microorganism must occur. If the unwanted microorganism grows, it signals a failure in the medium's selective components, and the entire batch must be rejected and investigated. Next, let's understand the method's suitability testing. This phase sets up the actual test by running 4 parallel arms. 1. Sample. The product sample is diluted and added to enrichment broth to encourage any existing microbe growth, then incubated and subcultured onto BCSA. 2. Product positive control. The product is inoculated with a not more than 100 CFU of the target organism to verify that the product matrix does not inhibit the recovery of the organism. This also proceeds to BCSA subculture. 3. Negative control, broth only, incubated, and subcultured. This ensures the media itself is sterile and free of contamination. 4. Positive control, broth inoculated with not more than 100 CFU of the target organism, incubated and subcultured. This verifies that the media and incubation conditions are capable of supporting growth. All enriched broths are incubated for not more than 48 hours at 30 to 35 degrees Celsius, and all subcultures onto BCSA are incubated under the same conditions for not more than 48 hours for final reading. Suitability of the test result interpretation. The United States Pharmacopoeia method for testing Burkholderia cepacia complex requires a method suitability study. This study ensures that the test is able to detect Burkholderia cepacia complex in the presence of the product. During this study, the incubation period must not exceed 48 hours, at a temperature of 30 to 35 degrees Celsius, because prolonged incubation can interfere with accuracy. Detection of Burkholderia cepacia complex is carried out on Burkholderia cepacia selective agar. On this medium, typical colonies may appear as greenish-brown colonies with yellow halos, or as white colonies surrounded by a pink to red zone. However, the appearance of colonies alone cannot confirm the identity of Burkholderia cepacia complex. Therefore, any growth observed on Burkholderia cepacia selective agar must undergo confirmatory identification testing, as described in United States Pharmacopoeia Chapter 1113, Microbial Characterization, Identification, and Strain Typing. The product is considered to pass the test if no Burkholderia cepacia complex colonies are observed, or if colonies that do appear are proven negative for Burkholderia cepacia complex after confirmatory identification. A failure is recorded only when confirmed Burkholderia cepacia complex colonies are present. Let's see product testing workflow. First step is sample preparation. Second step is primary incubation, and third is selective culture. Testing of products for Burkholderia cepacia complex. The testing procedure begins with the preparation of a test suspension of the product. A suitable amount of the product is transferred into a sterile container and diluted with an appropriate medium. Using a pipette, a measured volume of the prepared suspension is inoculated into soybean casein digest broth and mixed thoroughly. The inoculated broth is then incubated at a controlled temperature of 30 to 35 degrees Celsius for not more than 48 hours. After incubation, a portion of the broth culture is streaked onto the surface of a Burkholderia cepacia selective agar plate to allow for isolation of colonies. The agar plate is again incubated at 30 to 35 degrees Celsius for not more than 48 hours. Following incubation, the plates are carefully examined for colony growth. To confirm the presence of microorganisms, colonies that appear suspicious are further analyzed using confirmatory techniques. In some cases, a membrane filtration method can also be applied for microbial recovery, ensuring sensitive detection of any contaminating organisms. Result: interpretation. The growth of greenish-brown colonies with yellow halos, or white colonies surrounded by a pink to red zone on Burkholderia cepacia selective agar, indicates the possible presence of Burkholderia cepacia complex. However, the appearance of colonies alone is not sufficient for confirmation. Therefore, any growth observed on Burkholderia cepacia selective agar must be confirmed using identification tests. The product is considered to comply with the test if colonies of the described types are not present, or if the confirmatory identification tests give negative results. Despite all this, challenges remain due to unpredictable growth patterns, uneven contamination across batches. Resistance to preservatives survives even in antimicrobial formulations. False negatives due to delayed growth or insufficient detection time. So, never rely on a single test. Use layered testing and robust cGMP controls. Let's summarize. BCC is a resilient contaminant, thriving in water and resisting antibiotics. It creates a dual risk, harming patients and damaging manufacturing credibility. Prevention-first approach is essential via cGMP, validated methods, and regulatory compliance. Together, these ensure product integrity and patient safety. So, prevention, vigilance, and compliance go hand in hand.
Thank you all for being such an interactive and attentive audience. On behalf of Pharmaskillcraft, we appreciate your time and dedication to continuous learning. Remember, true pharmaceutical quality begins long before the final test. It begins with every clean pipe, validated method, and mindful professional. If you'd like to explore more courses, visit www.pharmaskillcraft.in. Stay curious, stay compliant, and keep raising the bar for patient safety.